I think that proteomics has come a very long way in improving the quantification of proteins and our confidence in the quantification of proteins. And we've done this while increasing the coverage, the number of proteins that we can see in the same experiment, at greater sensitivity, so we need less material in order to see all of those proteins. Throughput is still an issue, but we're getting better there. But to me, the one major achievement has been an improvement in the robustness and reproducibility of quantification. So the Clinical Proteogenomic Tumor Analysis Consortium and the International Cancer Proteogenomics Consortium are two organizations that have really done a lot to pull together investigators in the field of cancer proteomics across the US and across the world, and organize them around a similar set of questions with a similar approach, similar set of protocols, standard operating procedures, quality control metrics. This is huge because it now means that we can cross-reference from one experiment to the other and begin to build a very large database of information about the proteome of cancer, of many different types of cancers in many different countries, so we can look at the differences and the similarities. And the biggest sea change that enabled this was the culture of collaboration. The concept that you can work together with your frenemies, your competitors, on the same problem and work as a team, synergize your capabilities, not feel like you are competing, and a culture of data sharing and data access, so that whether you're a member of the consortium or not, you have access to these sets of data that you know were obtained with a certain set of protocols and a certain level of quality control, and you can start mining those data. And then instead of wasting money on repeating analyses over and over, generating data, you can do a better job of analyzing the data we have. We need to keep fostering that culture of collaboration. We need to stop rewarding investigators for being selfish and focusing only on getting there first. We need to reward them for sharing their data. There is so much information in the data that's being generated. There's 10 or 20 Cell, Nature, and Science papers there. We just need to continue to foster the climate of collaboration and of data access. Mass spectrometry is actually a relatively young science. And mass spectrometry applied to analyze proteins in clinical samples is even a younger part of that young family. There are growing pains. And 20 years ago, when we first started applying mass spectrometry to analysis of proteins, we tripped and stumbled quite a bit, like a toddler learning to walk. And some of the community hasn't realized how far we've gone and how far advanced we are and how mature the science is and how much better we are at reproducibility and robustness. The answer to my earlier question about what's the biggest advance in proteomics, and that is the reliability, the reproducibility of the quantification. That's really more important than sensitivity. People have to have confidence in our measurements. And people are just more familiar with antibody-based, and it's easier to implement. Mass spectrometry can be scary. Which button do I push? How do I inject? What parameters do I need to set? So I think it's still just a matter of evolution. I think technically we're still handicapped by slow throughput and by the expense and complexity of our instrument. But if we were to focus on targeted mass spec assays that addressed a very defined question, I have every confidence in the ability of engineers to build a black box mass spectrometer that could be as easy to utilize as an ELISA assay. But we need to know what assay we're trying to build. So there are a number of technical challenges yet in the field of mass spectrometry. Although we keep getting better at sensitivity, we keep getting better at depth of coverage, we keep getting better at throughput and speed, we've not yet integrated them all together so that we can really meet the clinical needs for throughput, sensitivity, and speed at the same time. And it's very much like the NASA statement, we can do it faster, cheaper, better. Well, in mass spec, we can do it more sensitively, more deeper, or faster, but you can only get 2. You can't get 3 at the same time. I think the hottest and most exciting area in mass spectrometry right now is single-cell proteomics. I've seen such advances in just the last 2 or 3 years in our ability to get thousands of proteins out of 1 to 10 cells. And this is going to enable spatially resolved proteomics, where you can go into a tissue section and really look at the epithelial component, the stromal compartment, the extracellular matrix, and the infiltrated immune cells, and get relatively deep proteomic and phosphoproteomic coverage on specific cellular compartments within the tissue. And I think that is the huge next step that we need to advance the basic biology. So it's always hard to be an early career person. The world is so intriguing. There are so many things you could do with yourself, and picking what to do is the hard one. And the biggest temptation that I see almost all young people doing is every idea is bright and beautiful, and I want to pursue every idea, and they're all equally important. And unfortunately, that's the recipe for failure, or at least for very slow advancement. You need to have a trajectory for yourself. You need to decide who you want to be known as, and keep your eye on that trajectory. You're going to be broad. You're going to have to use MALDI and ESI and all the different technologies in your field and be good at data integration. But think of a question that is a burning question for you that you want to answer, and put your energy into that. And in 5 years, you know what? You're going to be known as the person who really knows a lot about that problem and has a lot of different tools for solving it. Or you're going to be known as the person with a particular toolkit who can use it on a lot of different problems. But you need to be known for something. So figure out who you want to be and keep your eye on that path.